So we were asked to speak to the question of this issue of reproducibility in science from the funder's perspective, but I'm going to articulate that we're not just a funder, we're a foundation. We're interested in finding cures for patients. And these topics and these issues, they may sound like thought exercises and esoteric and checklists and all of the rest, but the reality is that this problem is a fundamental challenge that is affecting people's lives every day. And it has been the case for over 10 years now that we've been discussing this problem. And the question that I will challenge you all with is, are we really doing enough? I think this is a really important forum. It's a really important conference to be involved with. Our mission is to accelerate diagnostics and therapeutics for PTSD and traumatic brain injury. This is the trauma scorecard. What this is for PTSD, the number of approved diagnostics and therapeutics for treating these conditions, and for mild traumatic brain injury, the chronic effects of TBI. So you can see that the bar is very low for us to make progress in this area. And importantly, the reason we're not making progress in this area is are multifold. Partly, we don't understand the biology and the science sufficiently of these disorders. But when we started going into the root cause of why are we really not making progress, it really does come back to this issue of reproducibility, robustness, and rigor. And as a funder and someone who is investing in grants and science and the rest, we have to understand how good are the data that we're using to push forward an investment in any R&D enterprise. I come from the pharmaceutical industry. I was at Johnson & Johnson for over 15 years doing R&D research in translational medicine. I'm now leading this nonprofit foundation because I saw that we're missing some very fundamental platforms and infrastructure that are required to advance the field. And it wasn't obvious to me that those were being invested in by the government or industry. And sometimes the nonprofit sector is the place that does provide the infrastructure and can do this sort of thing. So that's what we do. We build enabling platforms with strategic partners, best-in-class partners, including industry, academia, foundations, to incentivize a team science approach. And most importantly, reproducibility, robustness, and rigor are at the core of everything we do. I'm going to describe some of the initiatives that we've been involved with over the last 10 years to do our little piece, to take action, and hopefully change and transform the paradigm of science, which needs to happen. This issue started long ago, 2005. John Ioannidis published this study, why most published research findings are false. I remember being at J&J and this paper coming out, and it was a big deal because as head of clinical programs, we knew that fundamentally this was true. But the academics and others that we were talking to just did not want to believe that this was true. This was just a spurious, interesting finding. But as then industry started to publish results showing that they too could not reproduce the science. And then finally NIH invested in their own efforts here and produced this NIH plan to enhance reproducibility, it became very clear that this isn't just an interesting observation, that there was a systematic problem in how we were conducting our science and that it needed to be addressed, which launched a number of initiatives worldwide, from the Society for Neuroscience, through the Wellcome Trust, and from our own preclinical data forum that we founded with the ECNP. This preclinical data forum was focused on neuroscience reproducibility, preclinical models, and it includes over 50 organizations, including academic organizations, pharmaceutical organizations, journal editors, and leaders from NIH and the IMI initiative. And we've published many papers. Early days, what were the root causes of these issues, and what are the problems that we need to resolve. And then we finally pivoted to what are we going to do about it. And we started putting together programming to address these issues, and I'll top line those. So in this preclinical space, sponsorship of projects to change the paradigm, the EQUIPPED project was funded by the IMI. The original idea for EQUIPPED, the original submission proposal, came from our Preclinical Data Forum team, and it was funded, and we are ourselves a co-sponsor of this along with many others. This is to address the quality of preclinical data collection and the science, fundamentally. We conduct multiple sponsored PDN workshops for young investigators. That next generation, how do we train them? Yes, you are doing all the right things. Yes, you should be lauded for that, and please don't stop. And we're trying to change that paradigm with the next generation through training programs that focus on what does reproducibility and rigor really look like? How do we even define those? And someone mentioned that earlier. That is still an outstanding question. We've published checklists for here, the consensus preclinical checklist to help guide investigators to know, well, do I have all the information I need to be transparent in my reporting and in my collection efforts? And within the last 2 years, we funded the first negative prize for negative scientific results. And the reason we did this was because we know that there is a lot of we'll call dark data out there, published results that are not making it to publication, negative results of legitimately conducted science, right? A trial that was done well, but the result was negative. And we wanted to really incentivize people to put that data out there just as readily as they put out the positive results, because otherwise the literature is extraordinarily biased to the positive findings. And if you're a funder, if you're someone who's investing in R&D, then you're getting a very biased view of the reality of the data and the performance of different molecules and systems. So we thought this was important to do. We thought it was just a $10,000 prize. We got an enormous number of submissions, and we had an awardee, and it was surprising how much interest there was in this prize. So we've re-upped it for this year, and we've expanded it to biomarkers as well. Within our organization, we launched the Amp It Up Consortium. This is another way to address the challenge, and this is asking the question, what does translational validity, construct validity mean between a preclinical model and the human disease condition, and how do we use these models more robustly, and how do we ensure that the data that we're collecting from them are useful, they have utility for informing these decisions. So this is the consortium brings together academics who are doing animal modeling, as well as clinicians who are doing the disease clinical studies. And pulling those two groups together in those first few meetings was really quite entertaining, because one side doesn't understand what the other side is dealing with, and what is meaningful, and how do we reverse engineer animal models to actually recapitulate the biology that we're interested in. We've also used this forum to industrialize animal models. So it's another way to deal with reproducibility. Maybe the labs do the primary science, but someone has to validate it, and that was not happening in the academic setting. So we worked with some of the leading providers of models and brought their models into an industrialized CRO setting. Psychogenics is a CRO that does preclinical modeling at scale, and we try to reproduce those methods and work with the investigators to establish whether these are translational models and reproducible models of biology. We've been working on this for 3 years. We have not yet been able to reestablish any of the 16 models that are out there for PTSD. We also believe that we need better tools. technologies to capture the data robustly, reproduce, and transparently. And we're partnering with IMEC, which is a nanotechnology consortium, and the Neuroscience Information Framework on testing the NeuroPixels probe for this particular area. Now, you may say, well, how is this reproducibility? Well, it's about the data, the quality of the data that we're getting. The preclinical animal studies are largely, a graduate student looking at an animal freezing or not freezing, and then judging whether and how many times that's happening, unless they've learned to use video. And even if they're using video, that video, how many frames per second is it collecting the data, and is it being reviewed through an AI mechanism? And if it is an AI mechanism, how good is that software? And so on and so on and so on. So technology really drives a lot of what we get out of the research, and we're trying to drive that frontier as well. And finally, we are absolutely interested in ensuring that data is shared in an open way. And for our needs, we decided that we needed to invest in a Brain Commons. This is next generation cloud-based infrastructure for data sharing of clinical as well as omic data, imaging data, neuroimaging data, and this does have the facility to start bringing in preclinical data. The key to all of this is to do this in a secured environment where the data are all graphed to a common data model, so that you can start thinking about integrating data across data sets, across cohorts, and being able to now scale the science, and doing this through the Brain Commons, which is based on an open source code model that the University of Chicago maintains, and working with the NIH on preclinical common data elements and instituting those in the data model are all really important elements. We use this Brain Commons to enforce our philosophy that if we're funding you to do this research, then you're going to share the results back with the research community. And so a lot of this is the carrots and the sticks. So what are the levers that we have as funders to enforce this? The RFA process itself, our first RFA for preclinical modeling, we gave feedback to the people who submitted their ideas to us. We told them why they didn't have a robust method. They were shocked and surprised that we were giving them that kind of feedback. Many of them took that feedback and incorporated it into their next submission to the NIH or to us, and then were successful in getting those awards. We blinded the reviewers to who they were reviewing. They didn't like it. They didn't understand why that was necessary, but what emerged was that what we funded was the best method, the best data, the best evidence, not whose name was on that particular proposal. Intramural experts on our side collaborate with the partner institutions and the grantees directly. Our team happens to come primarily from industry, and so we're able to provide industry-grade advice on how to do the data science, how to do the methodology at that level, diagnostics development, preclinical modeling, and more, and that's very helpful to them so that they can make their work translatable. We found very quickly that statistical analysis plans were absent in most of these proposals, and now we require that they are there and that they have to be developed up front. And we will even provide statistical support to them to develop those plans, because a lot of them don't have the expertise in-house or readily accessible, especially in the preclinical space. We provide tools. We provide the platform, the Brain Commons, for real-time data transfer. We ensure that we give them enough funding to do the data curation, management, upload, and it's obligated that they give us that data before they finish the program. So if at the milestones that they are supposed to give us the data, they don't get the next installment of funding. And that's pretty much the only way we get the data. And finally, we do believe that you need to invest in these consortia efforts, in these workshops, and education and training. Thank you.